Hi, Innerwheel. Innerwheel Australia has a unique history of generously supporting umbilical cord blood research, and my team has been lucky enough to be the recipients of Innerwheel support. I'd like to take this opportunity to let you know how your donations have influenced our research.
Umbilical cord blood is blood that's left behind in the cord and placenta after a baby's born. This blood is rich in hematopoietic stem cells, and the clinical potential of these stem cells has motivated the development of cord blood banks all around the world.
The stem cells from cord blood have potential clinical applications in the treatment of numerous diseases. But more importantly, these stem cells currently do have utility in the treatment of cancers such as leukemia and lymphoma. Stem cells from cord blood and their equivalent from adult bone marrow are the only stem cells routinely used in the clinic today.
Hematopoietic stem cell transplantation can be significantly improved through the provision of more stem cells, or else by improving the transplanted cell's ability to survive and engraft in a recipient's body. My work specifically aims to grow cord blood stem cells outside of the body, as well as priming these cells for improved engraftment following transplantation. The stem cells in cord blood are actually derived from the baby's bone marrow, and their normal function is to produce red and white blood cells throughout life. The anatomical location in the bone marrow where these cells are maintained is often referred to as the stem cell niche. Replicating this microenvironment is seen as one of the holy grails of medicine and stem cell biology. If we could replicate this microenvironment, we could grow cord blood stem cells outside of the body, and this would make cord blood transplantation safer and more effective. Currently, I am evaluating new cord blood transplant techniques using preclinical models. We hope that our studies will contribute to improved clinical outcomes for hematopoietic stem cell transplantations. This study is funded by Innerwheel Australia. Thank you, Innerwheel Australia, for allowing us to contribute to this field, and thank you to all the mums who have donated unbanked cord blood for research.
Prostate cancer is the second most common cancer in Australian men. If prostate cancer spreads, it most often travels in the blood to the patient's bone. Once the cancer forms a tumour in the bone, it becomes challenging to treat. Recent studies have shown that prostate cancer cells are attracted to factors secreted by the bone marrow from an area termed the stem cell niche. These same factors usually support cord blood stem cells. Our work builds upon knowledge gained from cord blood and stem cell research and combines it with prostate cancer research.
In fact, the effective replication of the processes of cancer spread in model systems is a necessary step in the development of mechanistic insight and development of new therapies. So we are working on the development of a bone marrow niche mimic to better understand prostate cancer metastases and to optimize drug treatments.
Similar to prostate cancer, breast cancer also has a preference for the bone as a metastatic site over other tissue. My project involves the development of bone marrow breast cancer metastasis models to understand how the bone marrow microenvironment influences breast cancer cell hierarchies and sensitivity to chemotherapies.
The placenta is rich in mesenchymal stem and stromal cells. Unlike the blood stem cells found in cord blood, which can only be used in specific patients, the cells found in placenta appear to be very versatile, like the cells from a universal blood donor. These cells secrete factors that dampen inflammatory processes and promote tissue regeneration. There are many potential applications for these cells.
A high percentage of bone fractures do not spontaneously repair, and some evolve into non-union fractures. We are developing a protocol to test if placental-derived stem cells, which are injected into fracture sites, can promote the repair of tissue through heart defects.
We're grateful for ongoing Innerwheel support for our cord blood research. Actually, growing cord blood stem cells remains a major goal in the field, and every advancement that brings us a step closer to safer and more effective cord blood transplants also brings us a step closer to understanding important processes in skeletal tissue repair and the spread of cancer.
Thank you, Innerwheel!